transferred and in what direction and in response to what kind of stimuli all remain relatively open questions. So during my postdoc, I've been using two approaches to study the function and dynamics of lipid droplets. And in one approach, I used a fluorescent fatty acid analog to follow the trafficking of fatty acids between lipid droplets and other organelles. And in the other approach, I was interested more generally in how these different contacts between different organelles are arranged. So we used multispectral imaging to label a number of organelles at once and then to see how they're organized in space and time. So for the first part of the talk, I'm going to be focused on this question of how do fatty acids transfer from lipid droplets to mitochondria during starvation? And this is work that I did together with Angelica Rambold, another postdoc in the lab who's interested in mitochondrial morphology and mitochondrial fusion. So this is important because cells need to be able to shift their metabolism in response to changing environmental or developmental conditions. Cells that are really growing and actively dividing tend to rely heavily on glycolysis, which is inefficient from an energy production standpoint, yielding only 2 molecules of ATP per molecule of glucose. But this provides a lot of building blocks for anabolic processes, for cell growth and division. When energy is more scarce, cells can shift to use oxidative phosphorylation, which produces 30 molecules of ATP per molecule of glucose. And if glucose is not available, then cells will use energy stored in triglycerides. So release fatty acids from triglycerides for transfer to mitochondria, where they undergo beta oxidation and then can then drive the TCA cycle. But some of the cell biology of how fatty acids traffic from lipid droplets to mitochondria wasn't clear. So this is what we set out to investigate. And to do this, I developed a fluorescence pulse chase assay using a fluorescent fatty acid. So this is a fatty acid that I'll call red C12. It has a Vodipy fluorophore and 12 carbons, but including the length of the fluorophore, it's more like palmitate, which is one of the most abundant fatty acids in the blood. So I feed cells with trace amounts of this fluorescent fatty acid overnight. It accumulates within lipid droplets, and then I can chase under different conditions, either complete medium or starvation, which in this case, this starvation is low glucose, no amino acids, and no serum. And then I can follow the trafficking of the fluorescent fatty acid. So here's what it looks like right after the pulse. All this fluorescent fatty acid has accumulated within structures that overlap really well with a lipid droplet marker. So all the fatty acid is in lipid droplets. And then if I chase for 24 hours in complete medium, the fatty acid stays in the lipid droplet. But if I starve for 24 hours, now this fluorescent fatty acid is no longer in the lipid droplets. And instead, it overlaps really well with a marker for mitochondria. So the fatty acid has moved from lipid droplets to mitochondria. And to validate this tool, we did some thin layer chromatography where lipids are spotted at the bottom of the plate, and then organic solvent runs up the plate, separating the lipids. So here's where the free red C12 runs on its own. And we can see that when we incubate it with cells, it's esterified, it's incorporated into neutral lipids. And if we starve the cells, we see these additional spots appear down here that can be prevented using the inhibitor etomoxir, which prevents transport of fatty acids into the mitochondria. So this is showing that this fluorescent fatty acid can be both esterified into neutral lipids and broken down, presumably through beta oxidation. So first we asked, how are fatty acids released from lipid droplets during starvation? And two different mechanisms have been described for how you can get fatty acids out of a lipid droplet. One is cytoplasmic lipases, such as ATGL is adipose triglyceride lipase, but it's actually a ubiquitously expressed cytoplasmic lipase. These lipases can sit on the surface of a lipid droplet and chew up triglycerides, releasing diacylglycerol and fatty acids. Or you can have autophagy of lipid droplets. In this case, this double membrane autophagosome structure comes and envelopes cytoplasm, including proteins, organelles, and then fuses with the lysosome, which delivers proteases and lipases. And so autophagy of lipid droplets has also been observed and is another way that fatty acids can get out of a lipid droplet. But which of these two mechanisms is used in response to starvation was not clear. So we first looked at the cytoplasmic lipases. So in using control siRNA, we see transfer of this fluorescent fatty acid from lipid droplets to mitochondria in response to starvation. But if we knock down ATGL, we no longer see this transfer. The fatty acid is staying in the lipid droplets. In contrast, if we use ATG5 knockout cells, which can't do autophagy, so I should mention that these assays are done in mouse embryonic fibroblasts because we have a variety of different knockouts that we can use. So in ATG5 knockouts, we still see significant transfer of the fluorescent fatty acid from lipid droplets to mitochondria. And we also looked at mitochondrial oxygen consumption as another measure of metabolism, and found that if we use a lipase inhibitor, it decreases the amount of mitochondrial oxygen consumption during starvation, while an autophagy inhibitor had no effect. So together, these are suggesting that during starvation, it's cytoplasmic lipases and not autophagy that are mainly responsible for releasing fatty acids from lipid droplets. But we wondered whether autophagy is playing another role in fatty acid trafficking during starvation. And the clue for this came from a surprising observation that lipid droplet number and size were actually increasing during starvation. So this is an inverted fluorescence micrograph looking at total lipid droplets. It's a hydrophobic dye labeling lipid droplets. And to our surprise, we found that when we were starving cells, rather than disappearing, the lipid droplets were actually, both the lipid droplet number and size were actually increasing up to 24 hours after starvation. But in ATG5 knockout cells, again, these are the cells that can't do autophagy, we didn't see this increase in lipid droplet number during starvation. So we asked, is autophagy refilling lipid droplets by releasing fatty acids from phospholipids within various membranes in the cell? And to test this, we used another variation of this, of a pulse chase assay using this time a fluorescent phospholipid. So here, this phospholipid has a fluorophore on one of the fatty acids within the phospholipid. So what we're following is this fatty acid that's within the phospholipid, in this case, phosphatidylcholine. And initially, after pulsing this into cells, it accumulates in a number of different membranes, including the ER, the mitochondria, and the Golgi. And then if we chase in complete medium, the distribution, again, doesn't really change. It's staying in the membranes. But if we starve the cells, now the signal from that fatty acid within the phospholipid is ending up in lipid droplets. And this depends on autophagy, because if we inhibit autophagy either by using 3-MA, which inhibits the upstream step of autophagy, or using baflomycin A1, which inhibits autophagosome-lysosome fusion, we no longer see this fatty acid trafficking to lipid droplets. So here it's staying in the membranes because there's no autophagy at all. And here these punctae are not overlapping. So the signal here is actually in autophagosomes. The fluorescent phospholipid is getting trapped in autophagosomes because it's not fusing with the lysosome. So fatty acids aren't being released and going to lipid droplets. So to summarize what I've shown so far, we found that in response to starvation, cytoplasmic lipase works on the surface of lipid droplets, releasing fatty acids for transfer to mitochondria. And meanwhile, autophagy of membranes in the cell is releasing fatty acids to refill the lipid droplets. So next we asked whether mitochondrial dynamics are playing a role.
Can I ask a question? Can you go back to the slide?
The previous?
Oh, yeah. The bottom one. Did you co-localize with LC3?
Yeah.
You did?
We did.
Okay.
So next we asked whether mitochondrial dynamics are playing a role in this process. And I'll tell you more about why we asked that in a minute, but just to give you a little bit of background about mitochondria. Mitochondria are really dynamic organelles that are constantly undergoing fusion and fission. Fusion mediated by MFN1 and MFN2 on the outer mitochondrial membrane and OPA1 on the inner mitochondrial membrane, while mitochondrial fission is mediated by DRP1. And my colleague Angelica, as well as Lucas Scorano's lab, had previously shown that in response to starvation, there's less mitochondrial fission. So overall, you end up with the balance is shifted towards fusion. So you get these really long tubulated mitochondria. But the physiological function of this mitochondrial tubulation wasn't known. So we asked, is this necessary for the metabolic switch over to metabolism of fatty acids that's happening in the cell? So we asked, what happens to fatty acid trafficking if mitochondrial fusion is blocked during starvation? Here's the same fatty acid trafficking assay that I've been showing, and but the scale here is a heat map showing the concentration of the fluorescent fatty acid, basically. So in wild-type cells, the mitochondria are fused in response to starvation, and the fatty acid is evenly distributed throughout the mitochondrial network. But if mitochondrial fusion is impaired, then some of the mitochondria get a really high concentration of the fatty acid, and some get very little. And we think this is because fatty acid transfer is happening at sites of close contact between lipid droplets and mitochondria. So in these mouse embryonic fibroblasts, there aren't that many lipid droplets. If the mitochondria are fragmented, then most of these mitochondria are not in contact with a lipid droplet. But in the wild-type cells where the mitochondria are fusing, more than 80% of the mitochondrial mass is continuous with a site of close contact with a lipid droplet. So we think that the fatty acids are transferring at these sites of contact between lipid droplets and mitochondria, and the mitochondrial fusion is necessary to distribute the fatty acid throughout the mitochondrial network. And we asked whether this distribution of fatty acids throughout the network is necessary for efficient metabolism. So it is. If we look at lipid-dependent mitochondrial oxygen consumption, we find that wild-type cells increase their beta oxidation about threefold in response to starvation, which allows them to maintain their total oxygen consumption at unstarved levels, at least for 24 hours. In contrast, the mitofusin 1 knockouts, which can't fuse their mitochondria, they are not able to upregulate beta oxidation, so their total mitochondrial oxygen consumption goes down. So what are the consequences of blocking mitochondrial fusion for the cell? In this case where the mitochondria can't fuse, some of the mitochondria are getting a lot of fatty acids, some are getting very little. This is resulting in inefficient oxidation. So what's happening to those fatty acids that aren't metabolized? One possibility is that they could be re-esterified and stored back into lipid droplets. So we do see that to some degree. The MFN1 and OPA1 knockout cells that can't fuse their mitochondria do have bigger and more lipid droplets during starvation than wild-type cells. But we weren't convinced that this could completely compensate for the lowered fatty acid metabolism. So we also asked, is it possible that some of these fatty acids get effluxed out of the cell? And to do this, we used a co-culture assay where we have donor cells incubated with this fluorescent fatty acid, And then we co-cultured them together with acceptor cells labeled in green and starved them to see if there's transfer of fatty acids between the cells. And in wild type, when the donors are wild type cells, we see some transfer. So here, this is an acceptor cell. It has the green label. It's outlined here, and there is some of the fluorescent fatty acid has transferred from donors to acceptors. But when the donor is a MFN1 or OPA1 knockout cell that can't fuse its mitochondria, this transfer is much more extreme. So we see a lot more transfer of fatty acid from the donor to the acceptor cell when the donor cells can't fuse their mitochondria. Okay, so to summarize this part of the talk, in wild-type cells, in response to starvation, what we see is that cytoplasmic lipases acting on lipid droplets release fatty acids for transfer to mitochondria. Meanwhile, autophagy of membranes is releasing fatty acids to refill the lipid droplets. And the mitochondria are fusing, which distributes fatty acids evenly throughout the mitochondrial network and allows for efficient metabolism of the fatty acids. In contrast, if mitochondrial fusion is impaired, then some of the mitochondria get a lot of the fatty acids, some get very little, and the fatty acids are not efficiently oxidized, so they're either stored back into lipid droplets or effluxed out of the cell, showing that this mitochondrial fusion deficiency is affecting not only this cell itself, but also the surrounding cells and tissue. In addition, another implication here was that this fatty acid transfer is happening at these contacts between lipid droplets and mitochondria. So for the next part of the talk, I'm going to talk more generally about the organization of different organelle contact sites in the cell.
Can I ask a question? So you're saying that when the mitochondria fragment, that basically you're not able to get the fatty acids to go basically from the lipid droplets to these other mitochondria.
They still get transferred. They still get transferred, but to the ones that aren't in contact.
Right. I was wondering if you could use like FRAP studies or things like that to sort of see whether or not like you bleach the mitochondria. It seems like there's some signal on those mitochondria.
Yeah. No, there definitely is some, but it seems like the transfer is less efficient.
Can I ask a question about the physiological importance of this? Like, what cells actually in the body have these droplets? Is it only the hepatocytes?
No, almost all cells, almost all.
And then what would starvation really be mimicking in this case, on a physiological level?
Yeah, well,
So when
I
did you
mean,
see
the
this transfer?
access of various cells to nutrients, it is going up and down, like with time of day.
Yeah, but it's not 0 versus 0, right? Or median.
Right. I mean, we're pushing things a little farther to see a phenotype, but definitely lots of cells are responding to changes in the availability of nutrients over the course of the day.
Are there any human diseases that can be associated with cell starvation, or can you relate it to any?
Well, in terms of like the mitochondrial fusion, the defects or mutations in mitofusins can cause Charcot-Marie tooth. So a lot of mitochondrial defects tend to cause.
Mitochondrial diseases.
Mm-hmm.
We kind of discussed this a little bit yesterday. So when do you decide, like, see, with the lipid group of fatty acids, obviously a pretty big perturbation to the fatty acid structure. Like, what do you sort of, like, what are certain things you benchmark to say, okay, this is really behaving like normal fatty acid, and what do you sort of like decide, okay, well, this is like, it crosses the barrier threshold of, okay, it's pretty good. What I'm going to conclude is like an authentic fatty acid.
Right. Good question. I mean, basically, in this case, we check that they're being esterified and they're being broken down. So, in that sense, we're reassured that they're still interacting with the enzymes that normally process fatty acids. But yeah, the kinetics are probably a bit different. But I think there are certain things. There are things that you can only do with these visual methods So it's always a trade-off, right? When in order to observe the system, you are going to be perturbing it to some degree.
Very cool. One question I have is, in a cell that's much more dependent on beta oxidation, so
Yeah.
which doesn't have this sort of starvation switch, but under normal conditions, keeps a lot of beta oxidation.
Yeah.
Are the kinetics the same? And the second part of it is, in a cell that's undergoing lipotoxicity, that has an overabundance of nutrition droplets, is there an actual impairment of the release of it, or is it a sense that it's being produced too much, or do you have any idea as to how those kinetics work?
Okay. Well, let me just answer the first question first. So definitely this is something that's really different in different cell types, and I think that would be really interesting to get into more going forward. So somebody that works on cardiac cells told me that with in the cells in the MEFs, when I do the pulse, I see the fluorescent fatty acid mostly accumulating and getting stored, put in lipid droplets, but they said they see it just go straight to mitochondria in cardiac cells. And then the second question was?
In lipotoxicity,
Mm-hmm.
is there too much autophagy? Do you have any sense as to is it an overproduction of lipid droplets, or is it a loss of release from the cell?
Right, great question. I mean, I imagine both could be involved, and I think these tools would allow me to get at some of those questions, but it's interesting.
Just ask one other question. So this would be different in different cells, and the cytosolic lipase that you're referring to would
Right.
presumably be different in different cells. What is this cell a model for, and what is that lipase then?
I mean, ATGL is pretty ubiquitously expressed.
It's in
So
liver and adipose.
other fibroblasts too. So, right. I mean, I guess the lab that I'm in now works under the principle that we're looking at things that all cells or most cells do, trying to find some common mechanisms. But I think, going forward, I'd love to get into into more different models, use more primary cells to see, because I think some of the players are going to be similar, but then the regulation will be very different in different cells and different tissue types. Okay, next part. So for the next part of the talk, I'm going to be talking more generally about how are organelles contacts organized in space and time, and this was done together with Alex Valm, who's an expert in linear unmixing algorithms for spectral imaging. So our goal here was to develop an imaging method to observe as many different organelle interactions at once as possible. And to do this, we simultaneously labeled 6 different organelles and then used multispectral imaging. So this is possible because a broad palette of fluorescent proteins have been developed here by Roger Chen's group as well as by others. And each of these fluorescent proteins has a characteristic absorption or excitation spectrum as well as a characteristic emission spectrum. So if we take 6 fluorophores whose emission spectra are as well separated as possible, there's still a lot of overlap. So there's no way to separate these fluorophores using filters. So instead, we used an approach. We started with a confocal approach using a confocal microscope with a spectral detector. So here, light that's emitted from the specimen is split by a diffraction grating onto a multi-anode detector so that we can collect an entire spectrum at each pixel in the image. And then we take a spectrum so here's a sample with a hypothetical sample with 6 fluorophores. Here there's a spectrum from 1 pixel. We take that and the spectra of the known spectra of the fluorophores that are in the sample, and we treat them as a system of linear equations. So we can use the method of least squares to find the best fit, and computationally we can determine the contribution of each of these fluorophores to that spectrum. And this allows us to distinguish fluorophores with highly overlapping spectra, even if they're present in the same pixel in the image. So here's what it looks like. This is a COS7 cell where the lysosomes, mitochondria, ER, peroxisomes, Golgi, and lipid droplets have all been labeled. So rather than the state of the art has been to look at 2 or 3 proteins at a time by lives for live cell imaging, and now we can look at 6 different compartments. And we use this to track lipid droplets. So in these movies, the lipid droplet that's being tracked on the left is outlined in white, and a blue line here means that the lipid droplet was successfully tracked, and a colored line means that it was within 1 pixel, or about 100 nanometers, of the indicated organelle. So most of these lipid droplets are spending a lot of time close to the ER, which is a major site of lipid synthesis. But other than that, they're doing all different kinds of things. So this one up here just makes a brief contact with the mitochondrion, while this lipid droplet down here is touching every other labeled organelle over the course of the 5 minutes. So we looked at the overall pattern of contacts between different organelles. So in this half matrix, each box represents a pair of organelles. This is lysosome Golgi, lysosome ER, and the scale is the number of contacts between those organelles. So, for example, between ER and mitochondria are the highest. And we were surprised to see that this overall pattern of organelle contacts was quite stable over time. So, I just showed you in the previous slide that the individual contacts are really transient and dynamic, but the overall pattern of contacts between organelles was really stable, at least over this 5-minute imaging period. As another way of quantifying this, we looked at the fraction of lipid droplets that were making contact with the different organelles. So at any one time, more than 80% of the lipid droplets are touching the ER, while about 20% are touching the mitochondria, and then fewer are making contact with those other organelles. And we compared, we also looked at 3-way contact. So contact, but this is a lipid droplet that is touching 2 other organelles. So in this case, a lipid droplet that's touching both a peroxisome and the Golgi, or both the mitochondria and the Golgi. And we compared what we observed with these ternary contacts to what you would expect based on pairwise contacts between the organelles. And some of these were as expected, while some were higher and some were lower. So we like this as proof of concept that we're getting information from this 6-color imaging that you couldn't get by just doing all the different pairs and trying to calculate how frequently the 3-way contacts would occur. And the one that was the lowest compared to expected was peroxisome lipid droplet Golgi contact, while the highest was lipid droplet contact with both ER and lysosome, which might indicate that the functions of these organelles are especially closely coordinated. So these contacts can shift in response to changing nutritional conditions. So in response to both starvation and excess fatty acid, we saw an increase in the contacts between lipid droplets and mitochondria, as well as an increase in the contacts between lipid droplets and lysosomes. So what I've been showing was confocal. It was 2-dimensional. To go fast enough to track organelles, we were just doing a single confocal slice. So next we worked with Eric Betzig's group at Janelia Research Campus to adapt their lattice light sheet microscope for spectral imaging. And in case you're not familiar with how light sheet microscopy works, one objective is